Hello, dear students. I am Dr. Sonal Mishra, posted in Government Digvijay PG Autonomous College, Rajnandgaon. Today, we are going to discuss about plant tissue culture. These are the contents we are going to deal today. Tissue culture. Tissue culture is a technique in which a small piece of viable tissue called explants are isolated from parent plants or mother plant and grown in a defined nutritional and controlled environment under aseptic condition, forming an undifferentiated mass of cell which is known as callus. Here in this diagram, this one is selected as a mother plant, explant is taken out from it and it is put in a defined nutrient media. There it grows and form callus. This callus will form embryoid which will develop into embryo and then whole plantlet will generate from it. Explant. Cell, tissue, or any organ of a plant which can be used to start tissue culture is known as explant. Any part of the plant can be used as explant for micropropagation. Explant. Here, this one is adult plant. We can select embryo, any of the organ, callus, or even a single cell and start the technique and process of tissue culture. All these will finally develop into a new adult plant. History of tissue culture. Some of the important contributions. First one is Haberland. He is known as father of plant tissue culture. He had first time suggested that a cell of a plant can be isolated and grown outside the plant body. Hanning, he has performed first embryo culture. Robinson Cote has done first root culture. Gottfried, White and Knopcott had performed first callus culture. Laurie has done first endosperm culture. Ranit and Stewart had performed somatic embryogenesis from carrot tissue. Maheshwari and Guha had done first pollen culture. Nan Yom had done first ovary culture. Tabaki et al. Had performed protoplast culture. Tissue culture technique depends on three fundamental abilities of plant. First one is totipotency. Totipotency is the ability of somatic cell of a plant to produce a complete new plant. That means from a single cell, the whole new plant can be regenerated. It implies that all the information necessary for growth and reproduction of the organism is contained within the cell itself. De-differentiation. Capacity of mature cell to return to the meristematic condition and development of a new growing point, followed by redifferentiation, which is the ability to reorganize into new organ. That means when already differentiated cells again become meristematic and redifferentiate themselves is known as dedifferentiation. Third one is competency, the endogenous potential of a given cell or a tissue to develop in a particular way. These three qualities help in the technique of plant tissue culture. The process of differentiation of different organs has named differently. Totipotent cell can be differentiated in many ways. If it form embryoid and then a complete plantlet, it is known as embryogenesis. When the process of differentiation of shoot take place, it is known as collagenesis, and differentiation of root from callus is called rhizogenesis. The cells of callus become differentiated to form xylem and phloem, then it is known as cytodifferentiation. The formation of root, stem, leaves, flower, etc. from the callus is known as organogenesis or morphogenesis. The formation of shoot, root, whole plantlet is known as colorhizogenesis. Aims of plant tissue culture: to develop new plant from the plant organ and seeds, to produce hybrid varieties of plant by combination of different genome from different plants, to produce disease-free plants, production of new plant varieties by somatic hybridization, to develop stress-resistant plants, to develop transgenic plant, to develop artificial seed, to develop hybrids. All these are the aims of plant tissue culture which can provide us benefit. Requirements for tissue culture. First of all, we will require a well-established laboratory, apparatus, nutrient media according to the plants we have selected, aseptic conditions, and requirement of light, temperature, and proper aeration is there. First of all, laboratory. For tissue culture, different rooms are required. Three clean rooms are required. First one is media preparation room, where washing, drawing, storage of vessels, and explant preparation is done. Where we perform preparation, sterilization, and storage of media. Second one is culture room, where aseptic handling of explant and cultures are done. Third one is growth room, where maintenance of culture is done and observation of culture is done. The apparatus we required are forceps, scalpel, autoclave for sterilization, pH meter to maintain the pH of media, weighing balance we required during preparation of media, micropipette, different needles, petri dish, and microscope, along with culture tubes, culture flask, hot air oven, UV lamp, laminar airflow, and inoculation chamber. Nutrient media. Several types of media had been prepared for the culture of cells and tissue, as different plant and different culture required different media selection. Different types of organic and inorganic substances are used to prepare these media. Vitamins, sugar, and some growth regulator like auxin, cytokinin, gibberellin, abscisic acid are added. Beside this, some extracts are also added like yeast extract, corn extract, malt extract, etc., which can improve the growth of culture and make it fast. All the components of the nutrient media are dissolved in distilled water, and agar-agar is added if there is need to solidify the media. The pH is maintained to 5.8. Now media is dispensed in flask and culture tubes, which are plugged with cotton, which allows the gaseous exchange and prevent the entry of microbes. Inorganic nutrients. In addition to carbon, hydrogen, and oxygen, all nutrient media provides 12 elements which are essential for plant growth. Macronutrient include sodium, potassium, phosphorus, calcium, sulfur, magnesium. Micronutrient includes iron, zinc, manganese, copper, boron, molybdenum. Some inorganic salts are also required like magnesium sulfate, potassium nitrate, potassium chloride, copper sulfate, etc. Sucrose is used as a carbon source, perchloride and EDTA as iron source. For vitamin source, thiamine, glycine, etc. are uses. Growth regulators are added like auxin, cytokinin, gibberellins, etc. Complex organic additives are also added to promote the growth of culture like yeast extract, coconut milk, corn milk, tomato juice, etc. Aseptic condition. It is generally seen that all the media have sugars, hence there is probability of growth of microorganisms on them which are harmful as they destroy the culture tissue by attacking them. Therefore, special precautions are taken before the media is used. For this, sterilization of living and non-living article is done. First one is sterilization of non-living articles, which we use like test tube, glassware, flask, beaker, anything we are going to use for tissue culture. After filling the culture media in the containers, cotton plugs are fitted tightly and autoclaved at 120 degrees centigrade for 15 to 20 minutes. Inoculation chamber before inoculating process is treated with UV rays to destroy microbes present in the environment. Now, sterilization of plant material. Firstly, plant material is treated with tipol for 5 to 10 minutes, thoroughly washed in tap water, then in distilled water. Then it is pushed submerged in 70% ethanol, then put into solution having mercury chloride 0.01%, 5 to 10% sodium hydroxide for 5 to 10 minutes. Shaken well to achieve 100% sterilization and then washed with distilled water to remove HgCl2. Light. For culturing of plant organs, the light having 2000 to 3000 lux intensity is required. When plants become capable of photosynthesis, it increases up to 3000 to 5000 lux intensity. Temperature. Tropical plants require relative high temperature than the temperate plants. Normally, 20 to 26 degrees centigrade is suitable for plant tissue culture. Generally, it is maintained to 25 degrees centigrade. Growth chambers are used to provide a specific temperature to cultured plant parts or plantlets. AC is required for maintaining the room temperature. Aeration. The tissue required to be cultured should get proper aeration. When the tissue are cultured in liquid medium, it is especially managed to enter it. For this, filter sterilized air is used. In addition, automatic shaker is used for entry of oxygen in the medium. Technique of tissue culture involves following steps. First one is selection of allied plant, preparation of culture media, initiation of culture, subculture stage, development and differentiation of subculture, shifting from test tube to fields. First one is selection of allied plants, or we can say mother plant. First of all, allied plant or mother plant is selected. The tissue and or organ of this plant should be used for tissue culturing. It could be a single cell, callus, protoplast, any organ, or we will go for micropropagation. Organ culture will include root culture, leaf culture, meristem culture, flower culture, anther culture, ovary culture, ovule culture, and embryo culture. Preparation of culture medium. Different plants require different types of culture media. For this, suitable culture media like white media, Skoog media, Miller and Skoog media, etc. is selected, and semi-solid or liquid culture media is prepared. Types of medium, it could be semi-solid, liquid, depending upon the concentration of agar. Initiation of culture. Maintenance of aseptic condition is the most important factor in tissue culture. For this, explant must be sterilized by chlorine water or mercury chloride solution. Then explants are inoculated under controlled temperature at 25 degrees centigrade in light. Subculture stage. After 2-3 weeks, the explant show visible growth by forming either callus or differentiated organs like shoot, root, etc. If large number of plantlets are needed from the callus, subculture of callus is done. The callus produced is taken out, cut into small pieces, and each piece is transferred to a fresh medium, or the callus in the medium is shaken vigorously, then it will separate into suspension and cell parts. Those cell parts are transferred to fresh medium in separate tubes where these pieces grow to form a mass of cell. This stage is known as subculture stage. Development and differentiation of subculture. The subcultured callus is proceed for further development and differentiation. If the concentration of phytohormones, that is auxin, gibberellin, cytokinin, are altered, then the callus can be induced to differentiate. A high cytokinin to auxin ratio induces shoot formation. That means if the concentration of cytokinin is high, shoot will form first. And a high auxin to cytokinin ratio induces root formation. That means if auxin is high in concentration, then it will produce root first. This is known as organogenesis. If the hormonal conditions are correctly balanced, then an entire plantlet can be induced to grow on culture medium, and this process is known as regeneration. Shifting from test tube to field. The test tube rooted plantlet are first subjected to acclimatization in greenhouse so that they can adjust with climatic conditions. Then they are shifted to field for further growth after field trials. Here in this diagram, the steps are represented. This one is allied plant selected, cell, tissue or organ is isolated and inoculated in a particular culture media. This one is medium, this is explant and this is cotton plug. In a span of time, this cell will develop into an undifferentiated mass of cell, that is callus. The subculturing of callus is done. Small part of this callus is taken out and transferred into another medium in another tube. This is the part of callus, cotton tube and the medium. Then the organogenesis can be generated through altering the auxin and cytokinin ratio. This shows the initiation of plantlet. New plant is formed and then it is potted in a pot. The whole plant is regenerated from a single cell, tissue or organ. This process is known as tissue culture. Types of plant tissue culture. Depending upon the explant selected, they are named variously. It can be embryo culture, seed culture, meristem culture, protoplast culture, organ culture, bud culture, callus culture, or even a single cell culture. First is callus culture. Callus culture is the culture of undifferentiated mass of cell. In this, an isolated piece of tissue, that is explant, taken out from a healthy plant and cultured on nutrient media, again to form a callus. When callus is grown in cultured media, many cells undergo division. Even some mature and differentiated cells like parenchyma, chlorenchyma undergo changes to become meristematic. This process is known as de-differentiation. Low light intensity and temperature between 25 to 28 degrees centigrade is required. This callus is then transferred to different media to regenerate a whole new plant. Here, this diagram shows callus culture and subculture stages. Explant placed in a media containing agar. This shows development of callus. This callus is shaken and form cell lumps and suspension culture. And when it is subcultured, the small part of this callus is taken out and transferred to different test tube, and this will again develop into a callus and form a new plantlet. This is known as subculture stage. Second one is single cell culture. The development of single cell on sterilized nutrient media in controlled environment. The callus is shaken in water to form suspension and separate single cells. The single cell is isolated with the help of needle and transferred on the filter paper placed on callus several days in advance. Single cell derive its nutrition from this filter paper, weighted by exudates from callus tissue. Though it is not in direct touch with the callus, the single cell start dividing to form colonies on filter paper, which are known as single cell clones, which are then isolated and cultured on nutrient media. This technique is called paper raft nurse technique. Here, this is a plant selected, callus is formed, suspension culture, single cell is isolated, it is placed in another medium, again callus is formed, this will develop shoot and root, new plantlet will form, and this is whole new plant regenerated from a single cell. This is paper raft nurse technique, callus, a single cell is taken out. This single cell is placed over the filter paper. This is the nurse callus. This single cell will derive its nutrition from this callus. This is the media. It will grow and form a new callus. This is known as single cell clones. One important contribution of tissue culture is formation of artificial seeds. Artificial seed can be defined as artificial encapsulation of somatic embryos. Somatic embryos or shoot buds are taken out from the mother plant and they are encapsulated to form artificial seeds. Artificial seeds have also been often referred to as synthetic seeds formed by encapsulating the viable somatic embryos in protective covering. The production of artificial seed is useful for plants which do not produce viable seeds. Artificial seeds are small size and this provide advantage of its storage, handling and shipping. The term embling is used for the plants originated from synthetic seeds. This is artificial seeds. It will behave similarly as a normal seeds. Protoplast culture. Protoplast is a naked cell surrounded by a plasma membrane. It can regenerate, cell wall, grow and divide. Protoplast is fragile but can be cultured and grow into a whole plant. It is a plant cell that has its cell wall completely removed using mechanical method or enzymatic methods. The term protoplast stands for a cell without cell wall. The fusion of protoplast of two different cells by spontaneous or induced method is called protoplast culture. Protoplast fusion technique can be used as a method to overcome the barriers of incompatibility and used for genetic manipulation of plant cell. This technique is used for combining distant genomes to produce somatic hybrids and hybrids. The protoplast culture is very useful in somatic hybridization and formation of hybrid, that is hybridization. Here this plant is selected, leaves are taken out, sterilized, removal of epidermis, released protoplast in enzymatic solution. Protoplast is washed, cell debris are removed, and we get isolated protoplast. Somatic hybridization. Development of hybrid plants through the fusion of somatic protoplast of two different plant species or varieties is called somatic hybridization. This involves fusion between isolated protoplast under in vitro condition and subsequent development of their product, that is heterokaryon, which develops into a hybrid plant. In this technique, the characters present in two or more species of plant is brought into a one single plant. Here this is a plant cell with a cell wall. Now it will treated with cellulase and pectinase. First we will treat it with pectinase to dissolve the middle lamella, then cellulose to dissolve the cell wall. Parent A, this one is protoplast of parent A, this one is of parent B. Now polyethylene glycol is used. The cell wall will fuse, dissolve and fuse. This is the nuclear fusion and we will get somatic hybrid cell. The process is known as somatic hybridization. Hybrids. Plant or cells which containing nucleus of one species but cytoplasm from both the parental species is known as hybrid. It is also known as cytoplasmic hybrids. For production of a hybrid, two parental cells are required, but there should be one complete cell, that means with cytoplasm and functional nucleus, and other should be without nucleus or inactive nucleus. Only cytoplasm fuse and genetic information from one of the two nuclei is lost. This is known as hybrid or cytoplasmic hybrids. That means this is a combination of cytoplasm from two different parental species, but nucleus from one single species. This one is plant A. This one is protoplast of plant B. Fusion will induce fused protoplast. Here the nucleus is inactive. The nucleus of protoplast A is eliminated and hybrid is formed. The hybrid protoplast is cultured and form a new plant. This is regenerated hybrid plant. Micropropagation. A variety of plant species can be propagated through the technique of cell, tissue or organ culture. This technique is known as clonal propagation or micropropagation. In most cases, it is done by placing sterilized shoot tips or auxiliary buds on a culture medium, which leads to formation of multiple buds. It includes following steps, that is establishment of tissue in the medium, formation of shoot, formation of root, then acclimatization in greenhouse in pots, and then transfer to fields after trials. Overview of micropropagation. This is the mother plant selected, callus is taken out or a single node or leaf. This will develop into embryogenesis by that is formation of embryos, shoot differentiation, multiple auxiliary branching, rooting in media, and this will produce adventitious buds. Finally, they all will produce a new plantlet. Somatic embryogenesis. The production of embryos from somatic or non-germ cell, that is other than zygote, usually involves a callus intermediate state. That means first of all callus is formed and then it forms a new plant. The embryos developing from zygote are known as zygotic embryo, while the other developing from pollen are known as pollen embryo or androgenetic embryos. Organogenesis. The production of root, shoot or leaves. These organs may arise out of pre-existing meristem or out of differentiated cell. This may involve a callus intermediate but often occurs without callus. Androgenesis. It is the technique for raising haploids from microspores or pollen grains. Generally, the whole anther is cultured. It includes the culture of anther or isolated pollen. Anther culture. This technique produces haploid plants. In this technique, floral buds are selected, sterilized with chlorine water, alcohol, and then dissected and placed in a nutrient medium with the help of forceps. This process is called inoculation. The petri plates are kept in optimum temperature as per the plant selected. For example, in tobacco plant, it is 5 degrees centigrade. This can produce 10 to 100 plants from one single anther. First, callus is formed by anther, which give rise to embryoids, and then a new plant developed from each embryoids. These are anthers isolated. They are cultured in a nutrient media. Either they will form embryoid and directly haploid plantlet, or first they will form callus and then they form haploid plant. Ovary culture. It is also termed as gynogenesis. Ovary is ovule bearing region which develops into fruit after pollination. In this technique, ovaries are isolated from pollinated or unpollinated flowers on the specific media to grow into fruits. This involves different stages with different requirements. Each stage has its own requirements. For example, in rice, two stages are recognized. First one is introduction. Here, ovaries are plotted on a liquid medium having low concentration of oxygen and kept in dark. Second step is regeneration. They are transferred to agar medium with high oxygen concentration and then kept in light. This is the diagram of ovary culture of Lycopersicum esculentum. This one is excised flower, culture of excised ovary, and a small fruit produced on culture. Embryo culture. Embryo culture is the culture of embryo excised from the plant ovary in early stage of plant development. They are surface sterilized and placed in water for few hours. Then they are placed on solid culture media consist of nutrients and vitamins. Now they are cultured at 25 degrees centigrade in dark until seedling grows up to 2 centimeter and root begins to emerge. Then grow in light until seedling has grown enough to be planted in soil. This technique helps to overcome problems related with embryo development due to incompatibility with the female tissues, absence of endosperm, long dormancy period of seeds, etc. Here, this is the embryo dissected after pollination, pro-embryo cultured on solid agar medium, plantlet developing from the embryo, plantlet transferred into soil in a pot. Seed culture. Seed culture is a technique in which a complete plant or seedling can be generated in vitro by taking the seed as explant. This technique is primarily used for orchids plant as the seed of these plants do not germinate well in vivo conditions. Factors affecting plant tissue culture. Selection of growth media. Minerals, growth factor, carbon sources, different hormones which we use for preparation of media. Environmental factors, availability of proper light, temperature, requirement of photo period, sterility, and media selected. Explant sources, usually the younger and less differentiated is the better explant for tissue culture. Different species show difference in ability to tissue culture technique. Advantages and application of plant tissue culture. Production of desired variation in plants. We can put all the good qualities in one single plant with the help of tissue culture technique. Production of disease resistant plant. Production of stress resistant varieties. Production of hybrid plants having good qualities from two different parents in a single plant. Production of artificial seed, as I had told you. Shortening of life cycle of plant. Production of new varieties of plant with improved qualities. We are going to discuss some of this application in detail in our next video. Thank you.